Hello everyone, my name is Jiwei Zhou. I'm a PhD researcher at Materials Experience Lab at TU Delft. I'm honored to present our paper, Living with Cyanobacteria, Exploring Materiality in Caring for Microbes in Everyday Life, on behalf of my co-authors Zjenja Doubrovski, Elisa Giaccardi, and Elvin Karana. This paper is about care for other than humans that we design and live with, and exploring the role of materiality in such care. The context of my research is microbial living artifacts, where living microorganisms provide humans with functional benefits such as purifying air. As microbes manifest their metabolic well-being at their unique timescale, designers often need to surface their livingness to human notice for timely care for these artifacts. In a previous work, we designed cyanochromic interfaces that surface the growth of cyanobacteria as fast color changes. We reimagined alternative reciprocal relationships between humans and microbes. What's also interesting in this work is the paperness of the interface, which we suggested to allow more intuitive, organic, and spontaneous care actions. The work has led to the research I'm presenting today. In this work, we ask how do people live with and care for a microbial living artifact. Furthermore, we look into the role of materiality in nurturing such care. In previous HCI works of microbial artifacts, care interactions are often prescribed by designers, difficult for creative configurations to emerge organically in the everyday. However, we think that the creative unfolding of care is important, referred to as improvisational haptic creativity in the book Matters of Care. It emphasizes uneventful everyday occurrences as transformative for humans to engage with more than human care as a way to disrupt the dominant anthropocentric view of innovation. We argue that materiality, namely material qualities of the artifacts, can facilitate such creative unfolding of care practices and thus a sense of cohabitation and co-evolution with other than human entities. What we are particularly interested in is the performative qualities of materials, which hold the potential to invite novel ways of doing. To explain what it is, in this example, the designer uses performative material qualities of foam, namely kneading and pressing, to evoke emotions of lingering longing for home and searching character of melancholy for expats. Another material quality we focus on is the temporality of cyanobacteria. They are ubiquitous photosynthetic microbes that have been used in living materials to capture carbon dioxide. While they function and grow well, they become greener over days and weeks. However, the change is too slow for human cohabitants to notice and provide timely care. Our study involves the design of cyanobacteria living artifact, paying special attention to its performative and temporal qualities. We then invited 8 individuals to live with and care for the artifact in their own homes for 2 weeks. So to bring cyanobacteria's livingness for human notice, we used a pH-indicating solution which signals the absorption of carbon dioxide by cyanobacteria during photosynthesis by changing color from yellow to purple in a matter of minutes. We jellified the cyanobacteria and the solution to form a material which could maintain its form and thus be easier to integrate into our artifact. Amongst the habitat elements crucial for cyanobacteria, we focus on light as the element that participants could modify through the artifact's performativity and multi-situatedness, because it is easily perceivable and adjustable within the shared domestic environment. Conventional options to maintain microbes alive, such as agar plates, are neither stable nor flexible enough to elicit from people the diverse creative performances we aim at. We explored alternative materials and developed an initial prototype with PDMS silicone rubber. It is robust, gas-permeable, adhesive, and transparent material that provides cyanobacteria with a viable habitat. We opted for an ambiguous and open-ended form, a simple sheet. I lived with and cared for the prototype for a week to evaluate its performativity. The adhesiveness and transparency effectively prompted light-altering care actions, but its size, thickness, and aesthetics hindered creative shaping and attachment. Our subsequent iterations prioritized reshaping the artifact to be more flexible and open-ended. We also added a darkened section to allow the avoidance of excessive lighting by folding the artifact. The living cyanobacteria artifact is a soft, flat, and rectangular object with a silicon outer shell. The artifact surfaces the photosynthesis of the microbe through color changes. It has a care label attached to the side suggesting approximate color references to the microbe's living states and a few care tips. We then invited 8 individuals living in the Netherlands to live with and care for the artifact for 2 weeks. They have various caretaking experiences for other than human living beings. They are microbiologists, plant lovers, kombucha maker, and even self-identified plant killer. Essentially, participants were directed to position the artifact in locations where air purification was considered necessary and light conditions optimal for its well-being. During the study, participants were prompted every 2 or 3 days to share their experiences. 2 interviews were conducted. Our interview inquiries focused on the evolution of care practices rather than the concept of materiality to mitigate potential biases and ensure accessibility of the questions. Our data was analyzed through 2 interrelated lenses: why and how of care, and how have the material qualities of the artifact influenced the care practices towards it. We've categorized participants' experiences into 4 themes. They are livingness, curiosity towards new life form, mutualism, and joyful interactions. I will mainly unfold 2 themes here. Curiosity towards a new life form motivated care. Participants encountered unique connections with the microbes and enjoyed their new responsibility. Although many struggled to align care actions with its well-being in the beginning, they could anticipate color changes when they repositioned the artifact towards the end of the study. Thus, a shift from excitement to relaxation was common. Towards the end of the study, a participant left the artifact in her corridor for the rest of the study, allowing it to do its own things. In parallel, they developed new care routines such as morning checks and documentation, and even imagined new practices beyond the current artifact. Mutualism is another motivation. A participant named the artifact Oxy, highlighting its perceived functional value. Some expressed doubts about the effectiveness of their air purification and desire for more tangible results. Interestingly, one participant took the artifact to their workplace, a beer brewery, intending to use it to absorb carbon dioxide while providing light for the microbe's well-being. We observed how material qualities of the artifact influenced care practices. The material composition effectively supported the viability of cyanobacteria. In 7 out of 8 households with sufficient lighting, care for the artifact was perceived as enjoyable. Participants appreciated the vibrant and ambiguous color change, indicating cyanobacteria's growth, well-being, and function, indicated as their unique language, while some expressed a wish for more immediate change to enhance their understanding. Participants explored various ways to place the artifact in the home. Towards the end of the study, many participants had evolved their own unique care practices, such as sticking the artifact onto a window for maximum light exposure or creating a light-receiving arc on their reading desk. Some expressed the reluctance to play with the artifact as it looks fragile. We identified 3 key roles that materiality plays in nurturing care practices and highlight design dimensions to capitalize on these roles. I will mainly unfold the 3rd role here. The study revealed that the artifact's performative qualities, like stickability, played a crucial role in encouraging people to actively explore various ways of interacting with living artifacts for both functionality and its well-being, leading to habitual care practices. Although the ambiguous form facilitated creative ways of care, designers can tailor the form for specific experiences, such as playfulness. Striking the right balance between open-endedness and specified guidance through form could cultivate creative care, sensibilities to microbes, while ensuring microbial well-being. Regarding broader opportunities and challenges associated with designing and caring for living artifacts, we call attention to the need in tuning into the evolving nature of care from improvisational to established, and call upon designers to be meticulous in conceptualizing the function of living artifacts, allowing mutualistic care relationships to naturally evolve within everyday practices of people who live with them. Finally, the study also suggested that good care is nuanced and subject to a wide array of interpretations among individuals. We extend an invitation to future research in the field of HCI to explore the intricate dimensions of care. For future work, the artifact can be crafted to be more sustainable regarding its longevity, for delving into the evolving of care practices over a longer period. Besides, it would be interesting to explore research methods that takes perspectives and agency of other than humans into account when exploring everyday interactions and exchanges. Finally, we suggest diving deeper into the tensions and dilemmas concerning functionality and care engagement. Ultimately, we aim to understand how human interactions with other than human artifacts can be enhanced to be more seamless, multidimensional, decentralized, and ultimately mutualistic. Finally, we'd like to highlight our contributions to the HCI community. Our study showcases a potential method for crafting habitats tailored to accommodate cyanobacteria and demonstrate its effectiveness in supporting longitudinal in situ investigations. We offer insights into their unique temporal patterns and behaviors within domestic settings. Furthermore, our work illuminates the pivotal role of materiality in the care of microbial living artifacts and highlights its performative potential as an important catalyst for HCI designers seeking to develop creative care approaches specifically tailored to these living artifacts. Thank you for listening.